Yeah, so this one? Yeah. I don't think yeah, so okay, go ahead. Go ahead, you go ahead with the introduction. Thank you so much.
So welcome to our first talk of the day. So it's my pleasure to introduce our first speaker of the day, Professor Jonathan Pettitt, who is a personal chair and professor at the University of Aberdeen, based at the Institute of Medical Sciences, which is just right there on the Prof Hill campus. Jonathan Pettitt started his academic career in biochemistry from Imperial College London. He then went on to do his PhD at Department of Pathology at the University of Cambridge, where he investigated the collagen gene structure and expression of the parasitic nematodes. And during his time at Cambridge, that's where Jonathan fell in love with the model organism C. elegans. And then this led to a postdoctoral fellowship across the continent in the US at the University of Colorado in Boulder, where he studied C. elegans development. He then continued this work at the Netherlands Cancer Institute in Amsterdam. And then Jonathan joined the University of Aberdeen in 1996 and has remained joint group leader of the Aberdeen Worm Laboratory, where he leads research on understanding nematode RNA splicing in the hopes of building therapeutics against parasitic nematodes that threaten human health and global food security. Jonathan is also current officer of public engagement with genetics of the Genetic Society UK, where he focuses on training young researchers to be better science communicators. And he has implemented most of this in his teaching at University of Aberdeen as well. He's also honorary curator for the University of Aberdeen's Museum and Special Collections. And Jonathan is also a recipient of numerous awards, most namely the prestigious JBS Pauline Award awarded by the Genetic Society in 2020 for his excellence in communicating genetics research to early audience. And that in particularly is due to his popular science talks on DNA ancestry and evolution. So it's my great pleasure to have Jonathan Pettitt here with us today. And his talk today is titled Understanding Human Ancestry from 60,000 years ago to today. And I hand over the floor now to Jonathan. Thank you very much.
Thank you, Adrian. Okay, so yeah, as Adrian said, I'm a bit of a fraud. I don't study human genetics. I study worm genetics. But one of the great things about genetics is it doesn't really matter what organism you study as long as it's diploid. The genetics is much the same. And so what I want to take you through today is just a very brief overview, obviously 60,000 years to today. What the explanation is, what does human genetic variation look like globally? And why is it the way it is? And it used to be our understanding of genetic variation, human genetic variation, was very much driven by medicine, okay? So understanding the genetic basis of disease, common diseases, as well as rare genetic diseases. That's changing, or has been changing for some time. You mentioned companies like 23andMe and Ancestry.com. People are now engaging directly with their genomes, okay? And so they're taking an interest in human genetics as a result of that. It's also being used to solve crime. I'm sure you all heard of the Golden Gate Killer, the first crime that was solved using genetic genealogy. And I am certain that there will be greater impacts as well. So it's important, and I take it as one of my missions, that people, there's a very good understanding, public understanding of human genetics. And it also has social impacts. Genetics has a very poor track record up until relatively recently for racism and eugenics. The founders of the field were literally eugenicists. And it's kind of ironic that modern genetics has shown that most of those ideas were wrong. Okay, so I don't know how many of you woke up this morning thinking about earwax, but you can divide humanity largely between whether you have sticky wet earwax and whether you have dry flaky earwax. Now this is a slide taken from 23andMe from some time ago. So there's a huge bias in individuals that have wet sticky earwax, and that's largely because there's a huge bias in 23andMe for Europeans. Okay. So what you're seeing there is a reflection of the data that exists in 23andMe. Primarily, individuals that have dry, flaky earwax are primarily living in East Asia. And we don't know why that is. Likely there is no reason for it. It's likely just a random chance that that's the way those variants are distributed. And it is a single nucleotide variant. Okay. The other thing I want to, when I've finished, what I want you to have an understanding of is something like this. So this is your typical readout for 23andMe for an individual. Again, it's taken from their website as an example. I don't know how many people here have done 23andMe or ancestry.com. Yeah, that's about normal. I certainly haven't either. Everybody's really surprised when they say you must have done it. It's like, no, I'd be really boring. I'd look like this. So the question is, well, what are those percentages? Right? How accurate are those percentages? What does this actually mean? And what are 23andMe doing when they do that, when they tell you that? So we're all aware, I'm sure we're all aware, that when we're looking at genetic variation, primarily it's not the only thing, but what companies like 23andMe and Ancestry.com are doing is they are looking at variants of a series of chemical letters, the 4 letters, A, T, C, and G, and they're looking for variants that occur in those letters. On average, any one of us, if I took any one of you and compared your genomes to mine, or if I took your 2 paternal and maternal genomes and compared them together, on average, you would find a difference in letters about once every 1,000 letters. Okay. We call those single nucleotide polymorphisms or single nucleotide variants. It's better if we stick with single nucleotide variants because it's confusing in Scotland because the acronym for single nucleotide polymorphisms uses SNP, and that can be quite confusing. So 23andMe are using an Ancestry, and these personal genomics company are using the pattern of single nucleotide polymorphisms to make the statements that they're doing. So this is an example. I won't use the laser pointer because it wouldn't work with it online. But this is an example of what I mean. Most people, you'll have, when you compare the 2 different genomes, they'll be identical, except for these points where we find these variants, these single nucleotide polymorphisms. And we can use these to define haplotypes. What do I mean by haplotypes? Well, haplotypes are blocks. The largest scale block that you can have is a chromosome. Okay? So by definition, you should have 23 different blocks, but it's not just as simple as that, because we also have to take into account the fact that we have mitochondrial DNA, which is inherited maternally, and we have Y chromosomes, which are inherited paternally. So everyone's genome will be a mosaic of their parents. But I'm sure you're aware, I hope you're aware, but if not, your genome will be a shuffled version of what was contributed to you by your parents. So Louise's genome in there, you can see that Linda has passed on one of her chromosome. Before passing it on, she shuffled her maternal and paternal chromosomes through the combination to pass that on. Whereas Bob over there has passed on that chromosome intact to Louise. Now, she hasn't inherited the Y chromosome, and she's inherited mitochondrial DNA from Linda. She'll also have got things that her parents don't have. On average, you will all be carrying 70 new mutations that were not present in your parent. Okay, so this is an ongoing event, but it happens because DNA replication is error-prone. Okay. So everyone's genome is a mosaic and everyone's genome is unique. You are quite by definition, at the genetic level, unique snowflakes. Okay, everybody is different. Your genome because it comes in these blocks that aren't just defined by chromosomes, they're defined by recombination. That's what defines your haplotypes. So what I'm going to do is we're going to start on a journey going backwards in time, and we're going to take a hypothetical situation where we go backwards up your ancestry tree. Initially, we're going to look at your genome just to see its behavior, and then what we're going to do is we're going to look at population effects, eventually going back to the foundation of humanity. So if you were to go backwards in time, and this is a slide taken from Graham Coop, so it's color-coded. So color-coded is the bits of the genome that you've inherited from your mum. Okay, or this hypothetical example is inherited from the mum. You can go further and go back up into the maternal grandma, and you can see it gets more fragmented. So what's happening as we go back in ancestry is your genome gets gradually more fragmented. Not everyone will be contributing equally either in your genealogy. Okay, so this is your genome in your mum. You can see, actually, that's really alarming. So in this particular case, the transmission of this chromosome 16, happened in its whole, but you've received nothing from chromosome 16 from your maternal grandma. Okay. We look in this chromosome 22, you see your mum passed it on whole. You received it from your maternal grandma, but your maternal grandma, before she passed it on to her daughter, your mum, underwent recombination events. So your genome, as we're going back further and back in time, will start to fragment, and this becomes important when we're talking about genealogy. So your genome as it exists now is 47 pieces of DNA. Okay, the chromosomes and the autosomal, the nuclear chromosomes and the mitochondrial DNA. On average, in your parent, it will be 47 plus 71, representing the recombination events. So already it will be fragmented. In your grandparents, it's 47 plus 71 plus 71. You can see the pattern that's emerging. This is an average because recombination has an element of randomness to it. What that means is if you go back in time, into your ancestors, it becomes smeared out. Okay, your very genetic identity becomes distributed across many individuals. You can see here, going back in time, in generations and in years ago. So by the time we get to 15 generations ago, so about 450 years ago, you've got about 1,000 or so ancestral segments, but they are distributed across 32,768 potential ancestors. Yes.
I'm a little confused. How did the hashtag ancestors be built from 4 to like 1,000?
That's the number of ancestors. Okay, but we're going to come back to that. Is there a problem?
Yeah, sorry, we get a comment from our Zoom attendees that they can't see the slides, so
Oh, that's
sorry.
a problem.
Please answer the question.
Oh, yeah, I'll answer the question. Yeah, that's a good point. So, as you go back in time, you've got 2 parents,
Yeah.
4 grandparents,
Yeah.
8 grandparents, and that will keep going.
Oh, it keeps multiplying by 2.
It will have to, right? Because you need 2 parents in order to have offspring. So it will keep going at each generation. We're going to come back to that because this is going to be important. Does that make sense?
Yeah.
Right. What that means is as you go back in your pedigrees, if you can go that far back, not everybody can. The Queen, for instance, can trace her ancestry. People have traced, or now the King, can trace their ancestry back to William the Conqueror through a defined route. At that point, though, the chances that William the Conqueror has given any DNA to King Charles is slim. Okay, because his genome would have exploded at that point, going backwards in time. So if we go back to what 23andMe is doing then, when they're giving you an ancestry pattern, they can't possibly be giving you an ancestry pattern based on going that far back in time. What they're doing is they're looking at individuals that exist in their database, and they're comparing your pattern of single nucleotide variants with the pattern of single nucleotide variants in their reference populations. Okay, now they have chosen their reference populations well. They generally choose global reference populations that have many generations at that geographical position. Okay, so there is a certain degree of accuracy, but when they are saying ancestry, it's important to be aware they can't be telling you explicitly about your ancestry, because some of that information as you go further back in time will have been lost. Okay, so next. But the other thing that we need to bear in mind as well is these sorts of patterns, they have a little bit of a downside, okay, because they tend to create this view of humanity that is discrete. So in other words, you can categorize people. I mean, some of these categories are ridiculous, for instance, ridiculously focused. You can narrow down even more. The other thing to bear in mind as well is this individual here is said to be 0.4% Japanese. What does that mean? Realistically, what that means is that's probably noise. One of the things that's always important to remember is ancestry companies, the techniques that they use are not 100% accurate. So when you're down in percentages like that, that's probably just error. Right. But the real problem is it suggests that these are defined populations. They're not. They're only defined in terms of the reference categories that people have used. The question then becomes is, well, how defined are human populations genetically? Okay, and so in order to explain a little bit more, we need to focus on what is it what is it that these ancestry companies are doing? They're not sequencing your genomes. Okay, you probably know this, but it's worth emphasizing that. What they're doing is they're using this thing here. Okay, this is the Illumina Global Screening Array. Okay, it's a chip. It's a microfabricated chip that allows them to take your DNA, the thing that you spit into the tube, when they isolate it, they put it onto this, and they assay your DNA for a surprisingly small number of single nucleotide polymorphisms. About 650,000. That's all they need, and that's largely because our DNA comes in these blocks. So the single nucleotide polymorphisms or variants can be used to define the blocks. You don't need to get all of them. You can just capture a subset. And these are defined from the 1,000 Genomes Project. Okay, so this was, as its name suggests, the sequencing. This was DNA sequencing of 1,000 genomes, some individuals from across the planet. And they use this then screening array to look for these SNPs. So why are they looking for these SNPs? What they're looking for are common variants that are found in everybody. If you think about what they're doing, you can't design a chip that can be used for everybody on the planet unless you're interrogating single nucleotide variants that are present generally in everybody on the planet. Okay, so by definition, they're using what are called globally common genetic variants. And how much of the human genome is that? Okay, so this is taken from a paper from John Novembre's lab, and it's one of the first attempts to present graphically what we've known as geneticists since the mid-2000s. Arguably earlier. And that is most of the differences between people on the planet, between anybody that you could care to choose, you and me, if you like, most of those differences are differences that are shared by between everybody. So they're differences between common single nucleotide variants. This is sometimes a hard thing to get your head around. The easiest way to think about it, one of the areas that was sampled in the 1,000 Genomes Project is Ibadan in Nigeria. specifically people of Yoruba ancestry in Ibadan in Nigeria. If you take two of those people and you compare their pattern of variance, what you find is that they are more different from one another than either of them is to somebody in France. Okay. So what does that mean? What that means is that there's more genetic variation if you're sampling in Ibadan in Nigeria than there is in sampling outside in France. Okay. And most of those variants are common variants. Okay, they're variants that are found everywhere on the planet in all these different regions: Africa, Europe, South Asia, East Asia, and the Americas. What's the patterns of variants that are geographically localized involve rare variants. Now, what does that mean? It means that if you're looking for geographically localized single nucleotide polymorphisms that are only found in that region, most people in that region won't have them because they're rare variants. Okay, so we've got a conundrum. How do we explain this? There's two things we need to explain. Why is most of the genetic variation in humanity, and it is most of it, greater than 50%, nearly, in some cases, depending on what we're looking at, it's 80%. Why is most of that distributed globally? And why are the ones that are only locally distributed very rare? Okay. And the answer to that comes from our unique demography. Okay. Humans are not genetically diverse for a global species. If we compare ourselves to wolves or crows, other species which have distributed themselves across the planet, we are much less genetically diverse. Okay. So why is that? I think I might have broken something. Okay. So this just shows from this data, this is taking all the polymorphisms that we can use and plotting them into the different categories. And it just goes to show that the dominant category is these common variants, and that yes, you can find geographically localized variants, but they are in that area very rare. Okay. The only example to that where you have rare variants that are found in two regions, in Africa and in the Americas, and that's because of recent transatlantic chattel slavery. Okay. So this is the reason why human genetic variation looks the way it is. We are a young species. Okay. Tops maybe 200,000 years. We all trace our ancestry back to a population that lived about 200,000 years ago. Consistently, the data shows that they lived somewhere in Africa. The evidence, current evidence, suggests that we are the unification of multiple populations that existed in parallel. But we do know that one of the pinch points in humanity, in modern humanity, was the out of Africa migration event, which I'm sure you have all heard of. Now that bottleneck, because it was an event where a smaller group of individuals left Africa. Now we don't know why that was. Okay, it might have been a Paleolithic gap year gone horribly wrong, but whatever it was, it has had profound effects on human genetic variation outside of Africa. It's reduced it. Okay. Which is why I can say, I told you about comparing two individuals in Ibadan compared to somebody from France. Generally speaking, the common genetic variation that we see outside of Africa is a subset of that which we see in Africa, which is what we would expect based on this pattern of migration. And each one of these migration events has created a bottleneck. So if we look across the planet, we'll see the last major bottleneck, the population of the Americas. If we look at individuals, indigenous individuals from the Americas, we find that they have the lowest level of genetic variation. Okay. Now, Now, the important thing to emphasize here is we are looking at fractions here. Tiny, tiny differences. We can see them because we're looking across the entire genome, but they are very subtle. And that's the key to these ancestry studies. You are looking at subtle variants. We are all, I mean, you commonly see people say, oh, we're all the same because we only differ by about 1 in 1,000 nucleotides. That's the thing, that we're the same, but we're all the same in that our differences are also the same. The regions that we differ in the same way. Okay. So effectively what you're seeing is we have frozen genetic variation. The majority of the genetic variation that you see in humans today globally is a freeze of the variation that existed in a population, our ancestral, this is freaky, it's like some sort of, anyway, ancestral population that existed at the point in which we had this out of Africa migration. So that's one explanation why human genetic variation looks like it does. But it's not the full story, because traditionally, people thought that humans could be divided, largely based on physical characteristics, into 5 major geographical regions. We still talk about them today. In fact, the 1,000 Genomes Project still groups people today. The Americas, Africa, Europe, East Asia, South Asia, almost as if these are defined groups of people. They are not. Okay? And the reason, but you would think, well, these events happened a long time ago. If these populations, let's say this population over here that moved to Africa, so Australia, bad geography, 50,000 years ago, if they stayed separate, you would expect to see patterns that were common in Australia and distinguish them from everybody else. But we don't. Right? That begs the question, well, why don't we? To answer that, we need to go back to our little exercise that I gave you at the beginning. Going back up your ancestral tree. Now, you highlighted this. Hang on a minute, you're saying, what's going on? You're getting large numbers? You are. You're getting impossibly large numbers. So this is your number of ancestors on the right there, growing as you go back in time. By the time you get to 1,000 years ago, you have potentially 34 billion ancestors. Now, that's unequivocal. You can't not have 2 parents at each point in your family tree. You have to have that. So what's going on? 1,000 years ago, we estimate that there was probably no more than 30 to 50 million people on the planet. Okay, so you've run out of ancestors. What's going on? Well, this is a simulation, again, started from Graham Coop, where he's taken an individual and again extrapolated back. And where you see the black circles are where an individual appears in this individual's family tree more than once. This is pedigree collapse. Okay? It's another way of saying is that if you go far enough in everybody's pedigree, you will find evidence of inbreeding. Now, this is Charles II. He's the Charles II of Spain. He's the poster child for inbreeding. He's the last Habsburg emperor in Spain. This is the pedigree that he's derived from. There he is at the bottom there. Don't do that, Jonathan. Each one of these yellows, not all of them, I haven't highlighted all of them, you can probably find others. Those are what's called inbreeding loops. The Habsburgs particularly favoured uncle-niece unions because that kept it all in the family. Yeah, everybody's done. Charles II is the output of this pattern of uncle-niece inbreeding. It took him, I think, till 8 before he could walk properly. He could never eat properly because his tongue was so swollen and his jaw was so deformed. As far as we were aware, he was sterile, or at least functionally sterile. and his body was racked by seizures, and he died just shy of his 39th birthday. Yes,
What does sterile mean in this case?
and he left no heirs. Okay, he produced no offspring, which is a bad thing if you're the Habsburg emperor. Okay, the fact that Charles II died early without heirs precipitated the Spanish Wars of Succession, in which thousands of people died. So don't tell me that genetics isn't important for history. But the point about this is that at the top of our trees, as we go back in time, we can look at this. It's an inevitable feature of our pedigrees. This is another example from Graham Coop simulations. In this case, we're taking two people, but it's the same thing as if I took, say, you and me. Okay, build our family trees backwards. Now the circles are saying how many generations back do we have to go before we have a common ancestor. In this particular case, it's 9 generations. Obviously, this is simulated data. What about real data? So I'm going to take you now through real data. If I can do this. Somebody's touched the screen, I think, so I can't. Just the next slide. We're back to next slide, please. Okay, maybe not. Doesn't matter. So if you're European, there we are. Because of the consequences of pedigree collapse, you're running out of ancestors. Okay. If you're European, by about the 13th century, 14th century, you have primarily European ancestry. So going back grandmother, great grandmothers, as I do. I don't know how many of you in the audience would fulfill that category, but if you do, that's the time that our common ancestor definitively lived. So there was somebody alive then who, if they left offspring, is definitively our genealogical ancestor. You might not ask one person. It's going to get better. 1,000 years ago, by about 1,000 years ago, everybody who was alive then is the ancestor in Europe, is the ancestor of everybody alive today in Europe. Or they're the ancestor of no one. What that means is that if you're European, all of our pedigrees converge 1,000 years ago. All of them. Right? That's how related we are, Europe. Okay. Let's not be Eurocentric, because we've had a bit of. Oh, sorry, yeah, that's the reason why I put this up. This is Abd al-Rahman III, this is the Caliph of Cordoba. He ruled Spain 1889 to 1961. Definitively, if you're a European, he is your ancestor. Definitively. Of course, the chance of inheriting any DNA from him, as I showed you earlier, is very, very slim. But he's your ancestor. Still not working. But what about globally? So there's a paper that was generated, a modeling paper in the 2000s, Douglas Road and co-workers, where they modeled the patterns of the expected patterns of interactions across the planet. And they were very conservative about how they did that. And their modeling data suggested that the time point which I said 1,000 years ago, everybody in Europe's pedigrees converge, have a guess what the global time point would be. About when in thousands of years ago? Okay. 6,000 years ago, between 6,000 and 7,000 years ago, everybody who was alive then is the ancestor of everybody alive now, or the ancestor of no one. That means all of our pedigrees are expected to converge about 6,000 or 7,000 years ago. Think about that. That's why we don't see any geographically localized common variants. We've never been isolated from one another. Now, when this paper came out, lots of people said, well, that's ridiculous. There are isolated communities all around the world. And their answer is, how isolated is isolated? We know that there was trade in Polynesia. Now, we know archaeological evidence that it was trade in Polynesia and the west coast of the Americas. Okay? In the Holocene, since the Holocene. We have never really been isolated. As Adam Rutherford said, we are horny and we are mobile. And that's all you need to make sure that you never get isolated populations. The point about Douglas Road and his co-workers were saying is you don't need these events to happen very often a century. Maybe once a century is enough over that time period to establish this flow. And obviously that the flow of information has only increased. So since the Ice Age, certainly, we have never been isolated as a global population. So the idea, this idea of that you'll see sometimes still today of racial purity, of people talking about, the genetics of a particular group, that's a myth. It's not borne out by the data. And in fact, as we sequence more and more data from individuals from across the planet, it's just reinforcing that. I'm just going to end. I realize I've probably gone too long. Still not working. I want to end with an example of this. So yeah, that's just for take home. So these people here. Okay, this is an example of members of the Fulani. Now the Fulani are spread pretty much all the way across Africa, sub-Saharan Africa, but traditionally and ancestrally they're pastoralists. Okay, so they, like most pastoralists around the world, they raise cattle and they drink milk. Now, in order to be able to drink milk as an adult and not experience problems, explosive diarrhea, anybody, you must be able to continue to produce lactase enzyme as an adult. Now, we're all familiar with Northern European populations being able to do that. And in order to do that, there we are. You must have a single nucleotide polymorphism. In Europe, it's this C residue here. And that C residue means that your gene stays on into adulthood, and so you can drink milk without any of the unnecessary effects. It's evolved multiple times. So we can see an example in Kenya, in the Bedouin population, and in Somalia. These are independent events. They all result in the same thing. We know that it's also evolved in South Asia as well, independently. In fact, evidence suggests that this is one of the strongest signals of selection in the human genome. We still don't fully understand why, but it's likely that it gives you the ability to survive starvation, periods of starvation. It's not under selection anymore if you have an industrial diet, okay? The key thing about this is that the Fulani have this. So they've acquired the European ability to drink milk as an adult. We think they acquired it about 2,000 years ago. So that's clear genetic evidence that there was population interaction between Europe, the European genetic signal coming from there, and sub-Saharan Africa 2,000 years ago. But that's just one evidence. It's ongoing and it's constant. Okay. Yeah, so this is my take-home message. Okay, if the human genome had a warning sign, it should be this. We have a shallow gene pool. Okay, we're surprisingly undiverse. What 23andMe and companies like that are doing, they are looking at the tiny, tiny fraction of variants that distinguish us. And most of those variants are ones that are shared across the planet. Okay, I'll stop there. Take any questions if I have time.
Yeah. So I don't know if it's Ancestry or 23andMe, but they advertised with that they can link you to some Viking genome. Yeah, like they had some like ancient genomes. Like,
Yes, that's right.
16, I don't know how many it was.
You can do, there's
Yeah.
a whole load, you can look at Neanderthals, I didn't talk about
Yeah, yeah.
Neanderthals, yeah.
And that's rubbish or?
No, it's it's not rubbish, and again, this will be a longer talk, but if I were to take in the archaic and ancestral genomes, what that data shows us very clearly is that you can find, I mean, they're our ancestors, so we should be able to see those genetic variants. But so, for instance, European populations, if you were to go back, let's go back 10,000 years. The individuals that lived here then were what we call Western hunter-gatherers. So they had, if our genetics is right, there are some issues with predicting, but if it maps onto how populations look today, they would have had very dark skin, dark hair, and blue eyes. They couldn't drink milk as an adult. Okay. The Western hunter-gatherers have made a contribution, but a small contribution to the modern European population. The bigger contribution was Anatolian farmers, which migrated in from about 8,000 years onward, lived in parallel to the Western hunter-gatherers. And then the next big wave was from the Pontic steppe, the Yamnaya, and they've moved in. Modern European populations are a composite of those three populations. Nobody today matches those populations. They don't exist in unmixed form. And that is true from every human population that we sample across the planet, okay? Going backwards in time, you start to see considerable changes. Populations that existed 4,000, 5,000, 10,000 years ago are gone. To answer your question, you can trace the genetics, you can see signatures. So you can say, oh, this particular segment of the genome, that's Western hunter-gatherer. But nobody is a pure Western hunter-gatherer. Sorry, I've probably gone on.
I just wanted to go to the also the 0.4% Japanese.
Yes.
And it said that's error. So what is it? Is it really error and they just wrap it as Japanese to make it interesting?
No, they don't. What the programs are doing is they're building up. So some of the percentages, like say, what percentage of your genome is matching this person that lives here? Built into that, there'll be some modeling going on in that. So it's not an unsophisticated approach, but you can think about, is that being called as a C or an A? If you get that mixed up, you might be saying, well, this it's used at this position. Most Japanese people have a C.
Oh, that's what. Okay, so it's not 100%.
But so it's just it's a variant calling problem.
Yeah,
and they should these exist.
And so one of the things that we hear with people that we work with, for instance, we have a big client in the Caribbean, who basically because they say, well, we're a melting pot here. So they're based in Bermuda, and so they have people from all over the world who came many generations. So they would like to understand their genetic setup.
Yeah.
Also for things like disease detection and prevention, because they feel that the human reference genome, which is European.
No, it's in the human reference genome. I think there are 9 individuals that are European, 2 individuals that are African American.
Okay.
But they were completely anonymous, of course.
Okay,
Nothing about them.
Yeah, so there's this idea of it being non-equitable. They say, well, we don't know whether we're properly represented here, and maybe there are certain kind of diseases that come out differently for us as they would come out for us.
So the key thing about looking at disease variation that you have to be very careful with, certainly in the US, there's a tradition of saying, if you have black skin, or if you classified as African American, we're going to test you for sickle cell anemia. Now, sickle cell anemia is a feature of being in the malaria belt. It's got no correlation, right, with skin color.
Okay.
And so one of the things that I think what human genetics would be promoting is you treat individuals on the basis of their genetics. So we could do that now. Like,
Yeah.
you don't need to. But you're right. Individuals that the product of recent colonialism will be a lot more, their genomes will be much more mosaic in terms of geography than you would expect.
Yeah. Interesting. Thank you very much.
Just my one above notes, I know it's a very famous case of Queen Victoria having hemophilia, because you mentioned sickle cell anemia.
Yeah.
And I know that she didn't have any children. So if she did have children, we the royal family nowadays might have hemophilia.
No, Queen Victoria had
Queen
to.
Victoria. She had some kind of genetic
Yeah,
disease,
haemophilia has been transmitted not into the British world family, it went into because we're talking about a royal family that was spread across Europe. So yeah, it did get transmitted. In fact, I think with the pedigree, you can see where the mutation arose.
It's interesting, because I read that online before, I found it very interesting.
The royals are good for genetics.
You mentioned something about Charles III of Spain. You said
Charles II, yeah.
he died without children. Does that mean he didn't have any children or he couldn't have children?
He had no children, and he did try with 2 wives. I mean, I feel sorry. I mean, we don't want it to be too hard on Charles II. It wasn't his fault, but the level of inbreeding that Charles II had, because you can build it from the pedigree, you can predict it with the pedigree, he was more inbred than if he was the offspring of a brother and a sister. So that's the level of inbreeding. As far as we're aware, he's the most inbred historical figure. Game of Thrones, there's a few people that are more inbred, but okay.
Thank you very much.
All right, thank you.
And thank you also to everyone online. I do apologize. You were the first one, Professor Pettitt. So we need to gear everything up. We're gonna make sure, because our next speaker is digital with us. So we're gonna make sure this person has all the proper rights to come in. So that might just give us a window of 2 minutes or so, just to buy a break, maybe get a coffee or something like that, while we do the setup correctly. And it is Dr. Who's our next speaker? Vinod Bhattarai on data in healthcare, exactly. So kind of not as much the genetics, but healthcare in the broader sense of the word. So if you just give us 2 minutes to clear that up, that would be much appreciated. You can recontinue on TikTok. Yeah, thank
I thought I'd make it a little bit more technical than a normal public engagement.
Hello, good morning, everyone.
Yeah, okay, good. And now
Hello, can you hear me?
Is this the first time you've been over here? No, this would be me. Yeah.
It's nice, so yeah.
It's nice. I mean, I've been past it. My GT goes on.
I don't know if there's a road for us to message.
Hello.
Yeah. Hello, sorry, we're just making sure you can, all the slides are visible.
All right, can I make just share my screen?
Yeah, just 1 second. We will tell you. So we now still have the presentation of Professor Pettitt on.
Okay. Sure.
Yeah. So, is there any way that we can find out, so for the people that are coming, how can we need to make sure that, so sorry, people are just having a 1 minute bio break, so everyone will come in and we will tell you when they're in, and then you can start.
All right, no worries.
Yeah. Oh, that's fine. Yeah, we can. Yeah, you can double check. Well, yeah. Yeah, that's a good idea. Maybe we should make videos. So you can navigate that from your laptop. This letting in because